Hello friends and welcome. In today's lecture we shall get an overview of lipids. We shall also try to classify lipids followed by underlying their functions. Let me start by the definition of lipids. A lipid is chemically defined as a substance that is insoluble in water and soluble in alcohol, ether and chloroform. Lipids are an important component of living cells. Together with carbohydrates and proteins, lipids are the main constituent of plant and animal cells. Lipids are the one class of large biological molecules that do not include true polymers, and they are generally not big enough to be considered macromolecules. The compounds called lipids are grouped with each other because they share one important trait: they mix poorly, if at all, with water. The hydrophobic behavior of lipids is based on their molecular structure. Although they may have some polar bonds associated with oxygen, lipids consist mostly of hydrocarbon regions. Lipids are varied in form and function. They include waxes and certain pigments. Lipids that are most biologically important are fats, phospholipids, and steroids. Now let's talk about the classification of lipids in detail. We'll talk about the classification of lipids based on their chemical composition, which includes A. Simple lipids, B. Compound lipids, C. Derived lipids. Simple lipids are homolipids. These are esters of fatty acids with various alcohols. First category, which comes under simple lipids, is fats and oils, that is, triglycerides and triacylglycerols. These are esters of fatty acids with a trihydroxy alcohol, glycerol. A fat is solid at ordinary room temperature, whereas an oil is liquid. Another category which comes under simple lipids is waxes. These are esters of fatty acids with high molecular weight, monohydroxy alcohols. B. Compound lipids or heterolipids. These are esters of fatty acids with alcohol and possess additional groups also. Compound lipids are broadly classified into two groups. First, phospholipids or phosphatids. These are compounds containing, in addition to fatty acids and glycerol, a phosphoric acid, nitrogen bases and other substituents. Second, glycolipids, cerebrosides. These are the compounds of fatty acids with carbohydrates and contain nitrogen but no phosphoric acid. The glycolipids also include certain structurally related compounds comprising the groups Ganglia Sites, Sulfolipids and sulfatids. Now, the third general class of lipids is derived lipids. These are the substances derived from simple and compound lipids by hydrolysis. These include fatty acids, alcohols, mono and diglycerides, steroids, terpenes and carotenoids. Glycerides and cholesterol esters, because of their uncharged nature, are also called neutral lipids. Now let's talk about them one by one. Simple lipids, fats and oils, that is, triglycerides or triacylglycerols. The triglycerides are the most abundant of all lipids. They constitute about 98% of total dietary lipids. The remaining 2% consist of phospholipids and cholesterol and its esters. They are the major components of storage or depot fats in plants and animal cells, but are not normally found in membranes. They are nonpolar hydrophobic molecules since they contain no electrically charged or highly polar functional groups. In animals, the fat cells or adipocytes contain very large quantities of triglycerides in the form of fat droplets, which fill almost the entire cell volume. Adipocytes are abundantly found under the skin, in the abdominal cavity, and in the mammary glands. Triglycerides can be stored in quantities sufficient to supply the energy needs of the body for many months, as in the case of obese persons. The Arctic and Antarctic animals such as whales, seals, walruses, and penguins are amply padded with triglycerides to serve both as energy storage depot and as an insulation against very low temperature. Chemically, triglycerides are esters of glycerol with 3 fatty acid molecules. The naturally occurring fats are of L type. Most of the triglycerides of nature are mixed or asymmetrical triglycerides. That is, they contain 2 or 3 different fatty acid units in the molecule. The term saturated fats and unsaturated fats are commonly used in the context of nutrition. These terms refer to the structure of the hydrocarbon chains of the fatty acids. If there are no double bonds between carbon atoms comprising a chain, such a structure is said to be saturated with hydrogen, and the resulting fatty acid is therefore called a saturated fatty acid. An unsaturated fatty acid has 1 or more double bonds with fewer hydrogen atoms on each double bonded carbon. Nearly all double bonds in naturally occurring fatty acids are cis double bonds, which cause a kink in the hydrocarbon chain wherever they occur. Moving on, let's talk about waxes now. The term wax originates in the Old English word wax, meaning the material of the honeycomb, reminding of beeswax the honeycomb is made of. These are esters of long-chain saturated and unsaturated fatty acids with long-chain monohydroxy alcohols. The fatty acids range in between C14 and C36, and the alcohols range from C16 to C36. In vertebrates, waxes are secreted by cutaneous glands as a protective coating to keep the skin pliable, lubricated, and waterproof. Birds, particularly waterfowl, secrete waxes in their preying glands to make their feathers water repellent. The leaves of many plants such as rhododendron, calotropis, etc. are shiny because of the deposition of protective waxy coating. Waxes also serve as the chief storage form of fuel in planktons. Waxes are unusually inert due to their saturated nature of the hydrocarbon chain. However, they can be split slowly with hot alcoholic potassium hydroxide. They are insoluble in water and highly resistant to atmospheric oxidation. Hence, these are used in polishing furnitures and automobiles, and also in wax-coated paper used to wrap perishable food products such as biscuits, cakes, etc. Now let's try to understand the compound lipids. In compound lipids, the first category is phospholipids or phosphatids. Phospholipids, the most abundant membrane lipids, they serve primarily as structural components of membranes and are never stored in large quantities. As their name implies, phospholipids contain phosphorus in the form of phosphoric acid groups. They differ from triglycerides in possessing usually one hydrophilic polar head group and usually two hydrophobic nonpolar tails. For this reason, they are often called polar lipids. Thus, phospholipids are amphipathic, whereas the storage lipids, triglycerides and waxes, are not. Because phospholipids have both polar and hydrophobic parts, when they are in water, they will spontaneously arrange themselves into ordered structures. In the cell, the most important of these is a bilayer. Water is excluded from the center of the bilayer where the fatty acids are, while the polar head groups have access to the water. Phospholipid bilayers form the basis for cell membranes and all the other organelle boundaries found in cells. Membranes found in cells also contain many other components such as free fatty acids, proteins, and other lipids. In phospholipids, two of the hydroxyl groups in glycerol are linked to fatty acids, while the third hydroxyl group is linked to phosphoric acid. The phosphate is further linked to one of a variety of small polar head groups or alcohols. Folch and Sperry have classified phospholipids into three categories, that is phosphoglycerides, phosphoinositides, and phosphosphingosides. Now let's talk about these types one by one. A. Phosphoglycerides. These are the major phospholipids found in membranes and contain two fatty acid molecules or tails esterified to the first and second hydroxyl groups of glycerol. The third hydroxyl group of glycerol forms an ester linkage with phosphoric acid. In addition, phosphoglycerides contain a second alcohol which is also esterified to the phosphoric acid. This is referred to as head alcohol group as it is present at one end, that is head of the long phosphoglyceride molecule. The various phosphoglycerides differ in their head alcohol groups. However, all of them contain two nonpolar tails, each consisting of a long chain, usually C16 or C18 fatty acids. Usually one of the fatty acids is saturated and the other unsaturated. The latter is always esterified to the middle of glycerol. Phosphoglycerides can be further divided into three types. These are lecithins or phosphatidylcholines. Lecithins, derived from lekithos, means yolk, are widely distributed in nature. Various oilseeds like soybean and the yeasts are important sources from plant world. In animals, the glandular and nervous tissues are rich in these lipids. The lecithins are required for the normal transport and utilization of as other lipids, especially in the liver of animals. On complete hydrolysis, lecithin yields choline, phosphoric acid, glycerol, and 2 moles of fatty acids. Second, cephalins. The cephalins are closely associated with lecithins in animal tissues. These are similar in structure to the lecithins, except that the choline is replaced by either ethanolamine or serine. Serine is the biochemical precursor of ethanolamine. Accordingly, two types of cephalins are recognized: phosphatidylethynolamine, and phosphatidylserine. Third, plasmalogens. Phosphoglycerol acetals, plasmalogens constitute about 10% of the phospholipids of the brain and muscles. Structurally, these resemble lecithins and cephalins but have one of the fatty acids replaced by an unsaturated ether. Since the nitrogen base can be choline, ethanolamine, or serine, three types of plasmalogens are accordingly distinguished: phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. Now, the second category in phospholipids is phosphoinositides or phosphatidylinositols. Phosphoinositides have been found to occur in phospholipids of brain tissues and of soybeans and are of considerable importance because of their role in transport processes in cells. These are phospholipids where a cyclic hexahydroxy alcohol called inositol replaces base. The inositol is present as the stereoisomer myoinositol. Depending on the number of phosphate groups, they can be mono, di, or triphosphoinositides. Third category of phospholipids is phosphosphingosides or sphingomyelins. These compounds are commonly found in nerve tissues, especially in the myelin sheath of the nerve, hence their name sphingomyelins, and apparently lack in plants and microorganisms. These differ from other phospholipids in their lack of glycerol and the presence of another nitrogenous base, sphingosine, or a closely related dihydrosphingosine, besides choline in place of glycerol. Sphingomyelins are electrically charged molecules and contain phosphocholine as their polar head groups. Now let's talk about another class of lipids, glycolipids or cerebrosides. They are also called as glycosphingosides. The cerebrosides, as the name suggests, are important constituents of brain where they amount to about 8% of the solid matter. These may also occur in tissues other than brain, since the head group characteristically consists of one or more sugar units. Cerebrosides are often called glycosphingosides. Both phospholipids and glycolipids form self-sealing lipid bilayers that are the basis for all cellular membranes. These are present in much higher concentration in medullated than in non-medullated nerve fibers. The structure of cerebrosides is somewhat similar to that of phosphosphingosides. They contain a high molecular weight fatty acid, sphingosine, and either galactose or glucose instead of choline, but no phosphoric acid. Moving on, let's talk about another category of lipids, gangliosides. These are found in significant concentration in ganglion cells of nervous tissue and also in most parenchymatous tissues like spleen and erythrocytes. They make up about 6% of the membrane lipids in the gray matter of the brain. Gangliosides are thought to act as receptors for toxic agents like the pathogens Vibrio cholera, influenza virus, and tetanus toxins. They are also implicated to play a role in cell-cell interaction. Next is sulfolipids. A glycolipid that contains sulfur is widely distributed in plants. It's localized in the chloroplast but is also found in the chromatophores of photosynthetic bacteria. Another category is sulfatides. A sulfate ester, analog of phrenosin, abundant in white matter of brain, is another sulfur-containing glycolipid. In it, the sulfate is present in ester linkage at C3 of the galactose portion of the molecule. Moving on, let's now talk about the final class of lipids called derived lipids. It includes the hydrolysis products of simple and compound lipids and also various other compounds such as steroids, terpenes, fatty acids, alcohols, fatty aldehydes, ketones, etc. Steroids. The steroids, steros means solid, are one of the most studied classes of biological compounds and are often found in association with fat. Since they contain no fatty acids, they are non-saponifiable. Fats, on the other hand, are saponifiable and form soaps when hydrolyzed with alkali. All steroids may be considered as derivatives of a fused and fully saturated ring system called cyclopentanoperhydrophenanthrene or styrene. This system consists of 3 cyclohexane rings A, B, and C, fused in non-linear or phenanthrene manner, and a terminal cyclopentane ring D. The next category is cholesterol. Cholesterol is not only an important component of some cell membranes and of plasma lipoproteins, but also the precursor of many other biologically important steroids such as bile acids and various steroid hormones. It occurs either free or as fatty esters in all animal cells. Cholesterol has a molecular formula C27H45OH. Another category is terpenes. Among the non-saponifiable lipids found in plants are many hydrocarbons known as terpenes. In general, these hydrocarbons and their oxygenated derivatives have lesser than 40 carbon atoms. The simplest terpenes are called monoterpenes and conform to the formula C10H16, which is equivalent to 2 isoprene units. Those with the formula C15H24 are called as sesquiterpenes, with C20H32 as diterpenes and with C30H48 as triterpenes. Terpenes with 40 carbon atoms include compounds called carotenoids. Terpenoids are made of varying number of repetitive units called isoprene units. Now let's study the functions of lipids. Lipids are stored in adipose tissue as triglycerides and are one of the major energy sources. Lipids are the good energy source as they provide a lot of calories as compared to carbohydrates. Some lipids are essential nutrient like fat-soluble vitamins A and D, vitamin E and vitamin K. During growth, they are utilized as brick for construction of biological membranes, so contributing to construction of the barrier which separates intracellular environment from extracellular one. Another function of lipids is many hormones are lipids. Steroid hormones like estrogens, androgens, and cortisol are formed from cholesterol, prostaglandins, prostacyclins, leukotrienes, thromboxanes, and other compounds from omega-3 and omega-6 polyunsaturated fatty acids with 20 carbon atoms. Many lipids like diacylglycerols, ceramides, sphingosine, and platelet activating factor act as regulators of intracellular processes. There are fat deposits not accessed during a fast, classified as structural fat, the function of which is to hold organs and nerves in the right position, protecting them against traumatic injuries and shock. Fat pads on the palms and buttocks protect the bones from mechanical pressure. A subcutaneous layer of fat is present in humans. It insulates the body, reducing the loss of body heat and contributing to maintain body temperature. Fats on epidermis are involved in maintaining water barrier. They are electrical insulators of axons of neurons, and the lipoprotein coating is called myelin sheath. Bile salts are natural detergents synthesized in the liver and secreted into bile. They solubilize phospholipids and cholesterol in the bile, permitting the secretion of cholesterol into the intestine. Bile salts also aid in the digestion and absorption of fat and soluble fat vitamins in gut. In many animals, some lipids are secreted into external environment and act as pheromones that attract or repel other organisms. They affect the texture and flavor of food and so its palatability. With that, we come to the end of today's lecture. Hope you have understood it well. Thanks and goodbye.